Welcome back to class, everyone. So we've been talking about different signal transduction pathways and mechanisms, how a ligand can bind to a G-coupled receptor or combine to a receptor tyrosine kinase, and how that can trigger cells and the messengers within the cell to turn on and off genes in response to the signal. Another type of really important signaling that happens is electrical and synaptic signaling in neurons. So that's what we're going to be talking about in chapter 22. I'm assuming you have had either a general biology class or an A&P class. We talk about nerve cells in more detail in those classes. This is just more of a review, really, about how they're signaling. So to begin with, this is a nerve cell that we're talking about, a motor neuron, or the nerves that control your muscles. They have multiple branched dendrites. That's all these little tree-like things here. And they have one single axon. It's longer than the dendrites, and it's more prominent than the dendrites. And the branches terminate in these little structures at the end called a synaptic bouton, or a terminal bulb or synaptic knob. You'll see them call these different things. At the point of that is where it's going to connect to the next cell to transmit the signal to the next cell. So your brain sends a signal, it goes through one cell to the next, to the next, to the next, until it gets to your muscle and controls your muscle, what it's doing. So the baton is what sends a signal to the next cell, or whether it's a neuron or a muscle or a gland cell to trigger that gland cell to release. Between two nerve cells, or between a nerve cell and a gland, or a nerve cell and a muscle cell, this junction between them is called a synapse. If we're talking about two nerve cells, here's nerves, here one nerve cell here, and one nerve cell here. This would be the axon of one nerve cell and the dendrite of another nerve cell. Although it could also be a dendrite and a dendrite connection to each other. Typically, neurons are going to make synapses with each other, with other neurons at the end of their axons and along their length as well, as you can see here. The signaling is coming down through this cell, and in the synapse, it's triggering and signaling the next cell to start signaling this signaling cascade. All cells, be it a nervous cell or another cell, has a membrane potential, or that change of positive and negative charges on either side of the membranes. Normally, there's an excess negative charge on the outside of the cell and a positive charge on the inside of the cell. That normal potential, it's called a resting membrane potential. And that potential can change. You can actually move the negative and positive charges on other sides of the cell to change that potential. One way we change a potential is just do simple diffusion. So it's going to go to diffuse from high to low concentrations. And if it's an ion, it's going to be diffusing through a receptor protein on a channel or a carrier protein. Because we have an uneven distribution of potassium inside and outside of the cell, that's a potassium ion gradient. And we keep that gradient because the sodium-potassium channel is constantly pulling potassium inside of the cell. And because of that gradient, potassium ions are accumulating inside the cell and are going to tend to diffuse to the outside of the cell where there's a lower concentration. Even though that solution as a whole is going to be electrically neutral, the charges can be locally separated. So you have a more positive charge on one side outside of the cell and a more negative charge on the inside of the membrane there. That's what gives you an electrical potential or voltage. And moving the ions is going to create a current. If someone's electrically excitable, such as your nerve cell, it can trigger changes in the membrane potential of that cell. And that change is going to create an action potential. During an action potential, you can see this quick little bloop up here and down. That's an action potential when the membrane potential is going to change from its negative to suddenly it's going to get very positive charge and then right back to negative again. Very quickly, less than a second that charge can happen. Our resting membrane potential is going to depend on our different ions inside and outside of the cell, and also on the selective permeability of that membrane to control which ion is able to move where. The side all in the extracellular fluid are going to have different kinds of cations and anions. Extracellularly, that fluid is going to have more dissolved salts and a lot of sodium chloride. And in the cytosol, you're going to have your trapped macromolecules, things like protein and RNA, that are too large to travel through a channel to get out of the cell. And there are two specific kinds of ion channels that are really important for keeping our resting membrane potential. One of them is a leak channel. Leak channels allow ions to leak across the membrane. They can be always open and allows potassium and sodium ions to diffuse in and out of the cell. The concentration of ion channels, how many channels are open, can affect how much of the potassium and sodium ions are able to go in and out. And that diffusion is going to depend on the concentration of the ion and the voltage of the membrane. There are more potassium leak channels than there are sodium leak channels, so potassium is going to diffuse more readily than sodium is. As the potassium ions, which are charged, are able to leave the side, all that's going to leave their anions, negative charge, is going to be left behind without their counterion. So we end up with this negative resting membrane potential inside the membrane. We're building up this negative concentration compared to the outside. I do want to emphasize, though, this isn't an ultimate charge so that it's ultimately negative or positive. It's a relative to each other. This has more of a cation, this has more of the anions, so it has a relative positive and negative charge. Another important pump in our charge of our membrane is a sodium-potassium pump. So the sodium-potassium pump is constantly pumping sodium out of the cell and pumping potassium into the cell. Compensating for that leakage, this ATP dependent, we're moving things counter to their concentration. On average, we're moving 3 positively charged sodium out for every 2 positively charged potassium moving in. So that's going to maintain a gradient. We're pumping out more cations and we're pumping in, supporting this positive charge on the extracellular side of the membrane and a more negative charge on the intracellular side inside the cell. Potassium is going to diffuse out of the cell, making the membrane potential more productive, more negative, and sodium is going to flow into the cell. That's going to drive the potassium, the potential in the positive direction, and it's called chlorine ions can also diffuse into the cell. But because of that negative membrane potential, those chloride ions are more repelled, so they're going to tend to more enter the cell along with positive ions. And making the membrane more permeable to chloride, increasing its permeability to the ion, is going to have 2 things to decrease the excitability of your neuron. One is you'll have a net entry of chloride ions without a cation, and you'll have what's called hyperpolarization. That means that your membrane potential is more negative, further away from sending a signal. And when the membrane is more permeable to sodium, some chloride will also enter. So a membrane that's permeable only to potassium ions, K+, will have a membrane potential that is equal to the equilibrium potential for potassium ions. So a potassium is going to come to an equilibrium, and if that's the only thing that can move across a cell, that's what's going to control the membrane potential. If the membrane is also permeable to a sodium, even just a little bit, the membrane potential can be partially depolarized as sodium leaks into the cell. And that entry of sodium is going to impact that electric potential, that movement of potassium. So potassium will be able to diffuse outward and balance the inward movement of sodium. Okay, that's an introduction to how we're holding a potential charge across our membranes. In our next videos, we're going to talk about how we change that charge, how that affects ourselves to our neurons, to send a cell signal and how that signal is picked up by other cells. See you later. Bye.